Hello everybody and welcome to the Functional Health Podcast with Ben Atkinson. This week I was delighted to speak to Professor Gordan Lauc. Gordan is a professor of biochemistry and molecular biology at the University of Zagreb, director of the National Centre of Scientific Excellence in Personalised Healthcare, honorary professor at multiple universities and member of the Johns Hopkins Society of Scholars. His research team is pioneering the application of something called glycans in the field of precision medicine and has developed a new form of biological age clock using these glycans. This adds to our arsenal in any attempt to improve our longevity and health span. This field is hugely fascinating. It's completely brand new to me, but I think you'll get a lot out of this one. Let's get into it. So, Professor Lauc, welcome to the show.
Thank you for inviting me. It's a pleasure to be here.
It's a real pleasure to have you on, sir. And it's lovely to have you on to dive into the fascinating world of glycans. I know this is something which is going to be quite new to a lot of people. So if we can start off with the bigger picture, and then we dive into understand how we can leverage glycans to improve our health span and longevity. So starting at the very beginning, what are glycans and what are their functions?
So if we go back a couple of billion years in evolution, when single-cell organisms were trying to compete with each other, avoid of being eaten and proliferate, they started to invent novel type of structures which will go beyond amino acids and proteins. So they started to add different monosaccharides to lipids and proteins and build glycoproteins and glycolipids. And today, the majority of proteins which were evolutionary invented after the life became multicellular are glycoproteins. So they have a polypeptide part, which is encoded by a gene. So we have a sequence of nucleotides, defines the amino acids, defines the polypeptide. But the majority of proteins also have a glycan part. So glycan part is being added post-translationally. It can even be modified later. It doesn't have a genetic template, but it's encoded in a network of dozens or hundreds of genes. So it's another layer of complexity which is determining the structure of proteins which perform all functions. So the glycans are kind of elaborate modifications of proteins which go beyond single gene, single protein paradigm. It's a network information in the dozens of genes, epigenetics, environment, determining the chemical structure of a glycan, which is being added to the protein. Maybe one good example are immunoglobulins, antibodies, proteins which are our major weapon in our arsenal against all the pathogens. And the so-called antigen binding fragment or the antigen recognition site is defined by a gene. So there is a gene sequence which determines what will this antibody bind. But at the moment when this gene is assembled or even fine-tuned, we don't know exactly what is the antigen. So antigen can be a virus, it can be a bacteria, it can be food. And the response of our immune system has to be very different depending on the type of the antigen. So the response of the immune system will be determined by alternative glycosylation, meaning putting different glycans to the Fc domain, which will then direct antibodies to the different effector functions. For example, if a specific glycan is added to antibody, it will activate natural killer cells and kill the target cell. But if different type of glycans are added to the same antibody, it will ignore the antigen and it will not kill the cell. So glycans enable proteins to become different later in life, to make decisions when they're needed. When we are infected, we have to make decisions. Because genes, we cannot change. We have our set of genes. They will change only by random mutations. And if these mutations happen in germ cells, then they will go to the next generation. And only through selection, we can modify the protein structure. So it goes very slowly. Glycans, we can change literally depending on what we eat or how we behave and so on.
So what you said makes perfect sense. So for example, would it also make sense that if there was kind of a dysfunction, would glycans mistake food or kind of glycosylate food in a certain way that the body would react to it as if it were a foreign body?
So there is one example, for example, one specific glycan structure which we do not have, which is N-glycolyl salic acid, is found in, for example, in red meat. And then we do treat it as a foreign antigen. And if we eat a lot of red meat, some of this structure can be incorporated into our cells, and then we consider our cells to be foreign, and then this could be responsible for the part of the autoimmunity. Mm-hmm. So yes, they can also be mistakes in glycosylation. But I think that the most relevant part is that we are all very different in the way we glycosylate our proteins. Even blood groups are chemically glycans. So the difference between A, B, and O blood groups is just different in a single monosaccharide on glycans. And there are many other examples how glycans makes us different, mm-hmm. which makes us as a population more resistant to any potential threat. So most pathogens bind to glycans. So whatever pathogen evolves, it will never recognize all humans. Just a subset of people will have a specific glycan structure. So it's a kind of a evolutionary tool to make us as a species more robust.
You mentioned in terms of determining our baseline of glycans and their function, and genetics is a major factor within that. Is there anything else, or are there any things that people should be wary of, like would have a certain genetic predisposition to have dysfunctional or maybe glycans which don't function as well as they could?
So genetics is generally, our genes is generally responsible between 30 and 50% of the glycan composition. So it is an important element. So all the sequence polymorphisms in our genes will explain up to 50% of variability in the glycosylation. But the rest is not encoded in a gene. It can be epigenetically encoded. So one of the most important information that the mother is transferring to their children in prenatal time is whether they are being born in a period of plenty, meaning there is a plenty of food, they have to be able to compete for it, they have to be strong, they have to be robust, or they're being born in a period of hunger where there is not enough food, so they should be as small as possible and try to survive until the better period comes. And this is encoded in our epigenome, and this is affecting also our protein glycosylation. And also the decisions we make during our life, depending like what we eat, how much are we physically active, even the way we think or how much we sleep, mm-hmm. will also affect protein glycosylation. So glycosylation is encoded partly in the genome, partly in the epigenome, and partly in environmental or metabolic factors which are integrating into the chemical structures which we can quantify as glycans.
Right. So that makes sense. And in terms of epigenetics, what actually happens? How do glycans... what are you measuring, essentially, is what I'm asking. So when, for example, someone has a disease, glycans obviously change to a certain degree. But how are you measuring that?
So there are multiple ways to quantify the glycan. Maybe the simplest way is you take a plasma sample. So you take blood, separate plasma proteins from the blood, just cut off all the glycans and analyze them and compare how many of each of the glycan structure is present. And mm-hmm. we know this changes in the disease, this changes with the changes in nutrition and so on. More elaborate way would be to choose a specific protein. For example, fish out IgG from the plasma, and then look only glycans and IgG. So you know these are the structures which are only found on IgG and not on other proteins. Mm-hmm. And we can go even deeper and look at the glycopeptide level, because IgG can be glycosylated on two different domains, and these glycans have different functions. And then you can look at the glycans on each on a glycosylation sites, which affect both disease predisposition but also disease course, because we have shown in several large studies that, for example, there are some glycans which are typical for people with diabetes, but we can see the change from the healthy profile toward the diabetes profile up to 10 years before people get diagnosed with diabetes.
Wow.
And these changes are gradual. So glycans gradually change through years as we are approaching the diagnosis, meaning our organism is trying to compensate for the damage which we are doing. And, the most of the diseases we have are the consequences of our lifestyle and the damage we do to our organism because we eat too much, we are not physically active, we are under stress, and so on. And glycans can quantify this change. And we have even shown that if you do something which is known to be healthy, for example, if you lose weight, extra weight, then glycans start moving backwards toward the healthy state. So it's a kind of a dynamic modifiable biomarker which is giving you a disease risk at a given moment.
Mm-hmm.
Because genes will give you disease risk at the moment of conception. This will never change through your life. But glycans are giving a dynamic disease risk, saying, oh, you are getting really close to the disease. You should really do something and try to fix it. And you try to fix it and you see whether it works or not.
That makes sense. I mean, this is fascinating because basically what you're saying, or what I'm interpreting you're saying, is that you can predict potential chronic disease before it happens.
Well, it's never absolute prediction. So you cannot take a person, measure a glycan, and say you will develop diabetes in 2 years and 5 days. You can't do something like that.
No.
But you can say, look, based on the glycan composition, you seem to be at a higher risk to develop diabetes in the next few years. And you do another test a year later, and you can say, look, now you're at even higher risk. Or your risk is now reduced because you have done something to reduce your risk. Mm-hmm. So it's, yes, you can see, because disease, what is a disease? So disease is a name which somebody 50, 100 years ago used to describe a symptom at a specific location. For example, you have inflammation in your joints. Oh, it's a rheumatoid arthritis. You have inflammation in your guts. Oh, it's a Crohn's disease. But the pathophysiological processes between these two diseases can be very similar. It's inflammation in both cases. And these inflammatory markers, which show that we are losing this compensatory ability, are visible for years before actually you get diagnosed.
Wow. Okay. That's amazing. I remember you published a paper, I think it was with David Sinclair, around with the IgG, glycan, and COVID, I think, looking at if these glycans are changed or there is
a... With David, we published more like an editorial saying, if you are concerned about the risk of severe COVID, then the biological age is more relevant than the chronological age, which we later also confirmed by studying the large cohorts of people, showing that, yes, if you have higher biological age measured by glycans, your risk to get severe COVID was much higher. And even we showed that during COVID, there is an accelerated aging. So when people get COVID, they get aged. Some people up to a decade within a week or two.
Wow.
People who
measured by glycans.
Measured by the glycanage, yeah. And the people who aged the most were at a higher risk to actually die. So kind of using up your rest of your life by a drastic viral infection. It's kind of a little bit spooky.
Yeah. I mean, that's fascinating, but it does make sense. I mean, some people obviously tragic died from COVID and therefore had a severe inflammatory reaction, but other people were asymptomatic and didn't experience anything at all. So you can understand why some people would be more affected from a glycan point of view than others. And additionally, due to the number of comorbidities, I think the average comorbidities was like 3 of the people who sadly died from COVID. that would affect glycan health as well. So biological age would be, for example, we know that people that have type 2 diabetes, or maybe they have, I don't know, COPD, for example, they will likely have a worse glycan health than those who do not have the disease. So therefore the biological age would be higher, and therefore the risk for COVID would be higher. So this, in my head, it seems to add up.
So it's kind of, so these glycans, which are part of this glycan clock of aging, are glycans which are regulating the function of immunoglobulins.
Mm-hmm.
One level of regulation of this low-grade systemic inflammation. And this is something what is underlying many different diseases. So actually, most of the diseases which occur late in life have an inflammatory component. So there is more and more inflammation going on in the body. And then the system will break somewhere. So it will break, if it breaks systematically, you have something like lupus. If you have some kind of other factor which will affect process in the gut, you will develop IBD. Also, there are some speculations that Alzheimer is a kind of a brain inflammation. So inflammation is one of the key problems we have when we live longer than it was kind of evolutionary optimized, because we were evolutionary optimized to live during our reproductive age. After we have transferred our genes, we don't have an effect on evolution anymore. So whether we live for 10, 20, 30, 50 more years, doesn't really matter genetically. And in this kind of overtime we all have now, because we all live much longer than 35 now,
mm-hmm.
inflammation becomes a major issue. Because obviously we don't cope well with inflammation as we are getting older. And we see a trace of this in glycans. So we can use glycans to quantify how pro-inflammatory a person is becoming. And this is correlated with different inflammatory diseases, including the different metabolic diseases which have this inflammatory component. Like, for example, diabetes also has an important inflammatory component.
And that's more type 2, which is more lifestyle.
Type 2, of course.
Yeah.
Type 1 is a different thing.
Yeah, I think it's important to make that distinction. Yeah.
But even, just the division between type 1 and type 2 is a very rough division. Diabetes is way more complex. There are multiple molecular pathways which can lead to dysregulation of glucose metabolism.
Mm-hmm. Mm-hmm. And in terms of modifiable risk factors now, so we know that you've alluded to certain diseases. I mean, that's not necessarily modifiable, but how do things like physical activity affect glycans?
This is actually, we have a very interesting story about that. So, we were kind of made believe that, you can sweat out the bad diet. You can eat junk food. If you exercise enough, you will be fine. And we had a large program. We had 1,000 people, middle-aged, couch potatoes, overweight, put in an exercise program.
Right.
After 3 months, most of them actually got worse in their glycanage. And now when I look backwards, when you start exercising, if you were not previously active, everything hurts. You have inflammation everywhere. Your feet hurt, your muscles hurt. So obviously there is more inflammation when you just start to be active.
Mm-hmm.
Then we said, okay, push them more. We have them to exercise another 6 months. Even after 9 months, most of them were still actually worse. So only when you have people who regularly exercise for years, you have a beneficial effect which you can quantify on this level of inflammation. But interestingly, there is another glycan which we have shown it's predictive of the future heart attack and stroke in women, interestingly only in women, and that one improved immediately. So some aspects of the glycom can be improved by physical activity more or less in any context, and physical activity is good. We need physical activity, but too much physical activity is not good. And I think this is a mistake many middle-aged people do when they just try to overdo it and fix all the problems in the last 30 years by running an extra hour a day or something like that. This does not work. So the physical activity has to be optimized and has to be tuned to a person, because we are all different, and somebody who is running for an hour every day can continue running for an hour, run an hour and a half. But somebody who was never running cannot start running an hour a day without having negative effect. And this is where glycan can help, because glycan can kind of monitor effects on an individual level. So I try something and then I check whether it worked for me. Because, when you think about medicine, we all talk about the personalized medicine, talk about pharmacogenomics, giving the right drug to the right patient. And this is accepted. But when we talk about the preventive medicine or nutrition, we don't talk about personalization. And here, personalization is even more important, because we are all extremely diverse. For example, if you think about the lactose tolerance, so most of Northern Europe would be lactose tolerant. So they were selected to be able to digest lactose even as adults. Most of Southern Europe and Africa would not be lactose tolerant. So they're lactose intolerant. So even though we are feeding on domesticated cattle for how many? 20,000 years, even more? It's only a portion of human population who adopted. And now think about the change in diet, which happened less than 100 years ago. So until 100 years ago, most of people were hungry, little bit of food. And now we moved into the period of plenty of food. Now the majority of population on the planet is overweight or obese. And we are not prepared for that. We don't have genes to cope with the fat cells which are here for 5 or 10 years. Fat cells never had fat for more than a year. Mm-hmm. So you accumulate fat and then you lose it in the winter. So there are no evolutionary mechanisms to preserve the fat cell to live for decades, or the glycogen. So the glycogen is a short-term storage of glucose, and it's supposed to be a buffer to enable kind of quick bursts of energy. And I think the majority of people on a planet are on a kind of a top glycogen storage all the time.
Yeah.
Because we eat all the time. So we always just store glucose, and then glucose has nowhere more to go. But also glycogen doesn't have a repair mechanism. So if there is a kind of damage on a glycogen, mm-hmm. It's damaged. And it can, and at the end, kill the cell, cause inflammation, and so on. And we are different in that way, because we know, for example, some people are considered to be happy, to be lucky, because they can eat, they don't get fat. Mm-hmm. It's kind of a good gene today. You can eat and you're not being obese. But these people would starve before puberty only 100 years ago. So, and then you give them all the same food and you give them all the same recommendation, which cannot work. So we have to find a way how to stratify people into different patterns of diet, different type of food. Mm-hmm. And when you start talking about food to people, it's like a religion. Somebody will claim, keto diet, you have to go for keto. You have to go for paleo. You have to go for low meat. And they're very firm beliefs. And these beliefs are fueled by some people who really respond to that food. And definitely, some people cannot tolerate a large amount of red meat. But there are some people who cannot tolerate a large amount of carbohydrates.
Or vegetables.
And are way better on meat. Mm-hmm. We need tools to see how to do it. Unfortunately, nutrigenomics is still not developed enough. When you think about all the polymorphisms, are there really any polymorphisms which can help us to guide our diet? Not really much. There's lactose, there's a coffee a bit, not so much. So what we can do, we can look at individual level and see the effects. So we had a study on 1,000 people, mm-hmm. 6 centers in Europe. They were put for 2 months on a low-calorie diet.
Yes.
Extreme low-calorie diet, 800 calories a day. And we looked at glycans before and after. and their glycans kind of improve. They become less inflammatory within 2 months. But nobody can live on an 800-calorie diet. You cannot give that recommendation. And then for the next 6 months, they were put on 5 different maintenance diets. So high glycemic index, high protein, low, different combinations. And interestingly, on each of the five diets, some people get better, some people got worse. So, there's no magic diet, nothing which works for everybody, but with the trial and error, we can find which one works for us.
Mm-hmm.
By looking at the glycans, because they're the ones which change first and they're easily quantifiable, we can relatively quickly see what actually works for me. Because, the only way we can benefit is to study ourselves, see how things work, try things, try supplements, try different dietary programs, and see what really helps you and what not.
Yes, absolutely. And I completely resonate with what you were saying about exercise and doing too much too quickly. I've injured myself because I went from weightlifting to running a marathon and then tried to go back to weightlifting as if I'd been doing it the whole time. And I ended up having a small tear in my bicep tendon in my shoulder. And I just knew whilst I was training at that level, coming back from running, that there was something wrong. Something felt off. I just felt inflamed all the time. And like you said, it probably just was too much too quickly and having more harm than good. And I think and as you get older, you have less resilience within that frame. So,
and problem is that everybody is different. You can have the best personal trainer in the world, but he will not know the difference between you and me, for example.
Mm-hmm.
And we can be very different. And if you go to guide for guidance, you'll get the same guidance. So it will have to be you at the end who will make decisions for yourself and say, this is good for me and this is not. And just blindly listening to the experts because they have a good advertisement, it's not the optimal way.
Mm-hmm. Yeah. Although the advertisement does help them.
Oh, of course.
And when you talked about food, drawing parallels, it's almost the same with food. Like if, for example, many people tout that fiber is very good for you. There's a lot of evidence to say that in some degree it is. It's not an essential nutrient, but there are some benefits to it. And people will go from eating something like 10 grams of fiber a day in total to 30 grams, which is the recommendation by Sakin in the UK. And what will happen is the complete opposite of what you expect. Either you will go to the toilet a lot or you won't at all. And you've just increased that too much too fast. And I imagine it's going to have not a healthy effect on you, but a bad one. And that is probably measurable by glycans as well, because it's going to disrupt your gut.
That's extremely complicated because you have to put in the microbiome in the context.
Yes, of course.
Because when you talk about the diet is heavily interplay between diet and the microbiome. Mm-hmm. And depending on the microbiome you have and the way microbiome is changing with your change in diet, you can respond in a different way. So I think that this whole fiber story is mostly about the microbiome. So we are actually feeding our microbiome, and then if we feed the right guys, it's okay. But you can give as much fiber as you want. If you don't have the good microbes, then they won't benefit from it. So I think to benefit from a specific diet, it has to be a combination of your genetics and your microbiome. Mm-hmm. Which then can be read out as a change in glycom. Because, for example, what we did, we looked into the glycom after the fecal microbial transplant. So some people have this transplant of the microbes.
Yes, for
pills or through some kind of colonoscopy, and then you have changed your microbiome, mm-hmm. and then we can see changes in the glycom. So your glycans in your plasma change if you change your microbes. So obviously there is a crosstalk.
That's fascinating. Yeah, because I've heard that being done for ulcerative colitis and indeed Crohn's as well.
Yeah, it's There and also Clostridium, some infections.
Yes, C.
So
diff.
and some people do it for fun. They want to have a healthy microbiome, so they take a healthy microbiome, which is, at the moment we don't know enough about microbiome.
Mm-hmm.
So we cannot make specific probiotic with these 25 strains which you want to take. So you just take, okay, these are the young guys. I take their microbiome, maybe it will get better. But in some cases, it really works. So it's, for example, in this Clostridium infection, it's more efficient than antibiotics.
Yes, I've seen the research on that. Yeah, I remember speaking to someone and they were saying that we might be taking poo pills sooner than you think, which is quite a funny thought, really.
I don't know whether you have a dog, but when I walk my dog, and if my dog is not feeling well,
mm-hmm.
he will eat all the poop there is around, because he wants to get all the microbes and change, obviously, some microbes in him which are not good.
Mm.
And all the animals, when they are babies, when they're growing, they're all surrounded by the feces. They get all these bacteria, which we don't do anymore. Now we think everything has to be sterile. No, don't get any bugs in yourself. And I think we are missing a big part of our ecosystem by missing all the bacteria which we used to have.
As a dedicated listener of my podcast, I'm absolutely sure you're familiar with my enthusiasm for preventative medicine. With preventable chronic diseases now causing 3 out of 5 deaths globally, we need to prioritize health management. Age is the most significant risk factor for chronic diseases, and while we can't alter our age, we can influence how we age on a molecular level, potentially preventing age-related illnesses. This is where the concept of the hallmarks of aging comes into play, with chronic inflammation being a key factor among the 12 hallmarks identified. Glycanage is the first biological age clock to measure chronic inflammation using glycans. Glycans are complex sugar molecules vital for cellular communication that can indicate the risk of 11 diseases up to 10 years in advance. But that's not all. Glycans also reflect your body's response to lifestyle changes, allowing you to actively manage your health. Technologies like these are poised to transform our approach to not only disease prevention, but your own health management, helping to make it more proactive than ever. Visit glycanage.com and use the code FUNCTIONALHEALTH at checkout for an exclusive offer. Take the first step towards a healthier future with Glycanage. Back to the show. Yeah, I think there is a lot to that. There was the hygiene hypothesis, where it's the idea that because everything is so clean, we don't get that immune tolerance, our immune system's not stimulated enough, so it wouldn't make us ill, but it would also train our immune system not to overreact to things in our environment, and therefore potentially reduce our risk of things like autoimmune diseases, which we know are on the rise now. So I completely agree with you. And interestingly, when you mentioned your dog, people that have dogs have a more diverse microbiome than people who don't. Same with cats.
Because you get everything from the dog. And people with dogs live longer.
Oh, I didn't know that.
Partly because they force you to walk. So I have to walk in the morning and evening. And also partly because of the microbiome.
Yes, yeah. And also they make you happy, right? Dogs are amazing.
Yeah.
Yeah, I had grew up with 2 Cocker Spaniels. What type of dog do you have?
It's a local hunting dog. It's called a tricolor Balkan hound.
Amazing.
It's a great dog. Stubborn. But it's a nice dog.
Amazing. I'm gonna have to Google that after this talk. 3-color Balkan. Okay. I want to loop back to exercise just for a second, because my brain was whirling a bit. Because obviously physical activity, what you do, whether you swim, run, cycle, or do weight training, surely that'll have a different effect on glycans. Do you know if that has been looked at before?
So we don't have enough studies to be specific.
Mm-hmm.
So when we had this 1,000 people training, we had several different exercise programs, and they all behaved similarly. But some of them responded positively, some negatively. So now we need some kind of a crossover study by changing the activity and see whether somebody who did not respond well for, I don't know, high intensity training, maybe endurance would be better for that person. We need to do more research. So definitely, glycan-wise, we are the largest lab in the world. We analyzed over 200,000 people so far, but 200,000 people is not that much. We need to do millions to get all these details about the types of activity and duration and intensity and combination with the genetics and the age. It's really complicated. So kind of my feeling at today's level of knowledge and what can be done, the glycans are most helpful in kind of monitoring yourself. I measure my glycans every few months, and then I can see effects of infection, gaining weight, losing weight, and so on. And it's a kind of a good feedback for the long-term changes. Mm-hmm. Because we all need feedback. For example, I learned that I have to have a continuous glucose monitor, and I have it most of the time, because otherwise I eat when nobody is looking. No, there is food around, or I eat something. Just grab something and eat it.
Yeah, absent-mindedly, right? Sometimes people don't realize.
I know. If I know, oh, there is something here, but if I eat it, I will see that spike, and I don't want to see all these spikes. So then I say, okay, I'll make my periods of not eating longer. So, we need the feedback. And this continuous glucose monitors can give us a quick feedback. So, what I did in the last few hours. And glycans give you feedback what happened in the last few months.
Mm-hmm. What has been the biggest lever that you've pulled to improve your glycan health or glycanage?
I tried several things. I'm not really persistent. So I probably have two major issues in my life. I eat too much carbohydrates, definitely. And I try to reduce them, but for a short time it's working. Then I start eating them again. And the other problem which I identified was some kind of cryptic infections which were going on. For example, I have a problem with one of the root canals, which I was not aware of until I started to systematically look, okay, where is inflammation in my body coming from? And then I thought, okay, there are some bugs growing here that I was not aware. When this was taken care of, even the glycanage started improving. So glycanage can also be indicative of this chronic infections which are going on somewhere which you are maybe even not aware of.
Yeah, that's fascinating. I mean, I think people, because sometimes root canals can go unnoticed for a very long period of time, right? And that causes systemic inflammation. Yeah.
But, it's practically, it's war. It's bacteria, immune system coming in, fighting. And the fact that this root the nerve was first removed, so you don't feel pain, you don't get the feedback. Mm-hmm. So, it's not killing you, and your immune system is still strong enough to contain it, but it's wasting your resources and it's making you more pro-inflammatory, and then you would respond to some other inflammation somewhere else in the body more aggressively because you have a war going on.
Yeah, that's true as well. And I guess if there was ever a case, and touch wood, it doesn't happen. But if you were immunocompromised, then it becomes more of an issue than it.
Then it becomes.
Yeah. Moving on to another risk factor, which I think many people probably don't think affects their longevity, is stress. Is this something which has a direct effect on glycans? And noted there are many different types of stress, good stresses and bad stresses.
So stress is a very difficult topic to study. Mm-hmm. We tried a lot, and actually even for 30 years I was doing some kind of a stress research. And the problem is that it's very hard to get objective data from the patient, meaning what is really stress, how do you quantify it? Mm. Is it external stressor or response to stress? So there's very little really good studies on stress. We did something on animals where we have very clear-cut results. Yes, stress is changing your glycans. And kind of from doing the glycanage and having a large number of people tested, we know that busy executives are often not that good. So obviously, stress is a major factor. Whether it's a directly stress affecting the immune system, which we know it happens,
The
or is
cortisol.
it just stress changing your lifestyle habits and dietary patterns, exercise or not exercise because of stress? I don't know. But definitely the way we think and the way we cope with life is reflected in glycans. So you can definitely, we can see a busy, non-compensated executive, we can see it in a glycom. But again, we have examples of people who are at extremely high levels in either industry or government, and some of them are coping really well. So it's really individual.
I mean, I think some people are just built differently. You know, I think the Elon Musks of the world, where he will outwork everyone and just has a level of resilience which maybe most people do not, certainly more than I. And it doesn't seem to affect him as much as others. When I think...
We didn't test him.
Yet.
Some other of the major corporate CEOs in US, we didn't test him. You really don't know. Because, just by looking at a person, you don't see what is really inside.
That's true.
So, for example, I'm a very calm person. It's very hard to kind of shake me, but it doesn't mean that I'm not responding. I'm just not showing the response. So it's and sometimes people who have very aggressive externalization of stress, it might be a better way of coping than just pretending nothing happens. So it's, I wouldn't say that we are built to sustain stress, because we are not. Stress is a fight or flight response. It's a very short-term adaptation to danger, which is increasing your chances of survival. At the moment, the stress which is causing this response is not the real danger, meaning somebody wants to eat me, but it's any kind of a life situation which is common today. I'm late, I have a horrible boss or whatever. There has to be a negative physiological effect, because it's activating the stress response, it's shutting down the immune system, it's shutting down the digestive tract, reproductive tract. Stress is not good. But external situation does not translate to the stress response linearly.
Yeah. Yes.
Some people can be in the worst possible situation and just accept it, and the other people can feel horrible because maybe they will die in 2 years because of a meteor attack or something like that. So it's all in our heads.
I think those people are stressed all the time, the latter, I mean. But you're absolutely right. Your perception of the given situation can completely affect your physiological response to it. So we also talk about like the HPA axis, so hypothalamic pituitary adrenal axis. And you can measure the cortisol, kind of the increase, the ebb and flow of it throughout the day. And in the morning, it increases rapidly. But this will be different for different people in different circumstances. And if it's dysregulated, people will spike later in the evening. So they might not get it in the morning, feel really tired, unable to get out of bed very well. And then they'll have energy at like 7 p.m., which isn't useful for sleep. And it's like a vicious circle.
The cortisol and all this complex of its receptor and all the proteins which come together, and so many come in, so many come out. It's so individual. It's so depending on past events, on epigenetics, on nutritional status at the moment. It's really extremely individual. It can't be generalized.
Yes, I completely agree. And that's why I was just trying to echo what you were saying, that stress, you just can't measure it, or you can't just look at someone and say one person's more stressed than the other. For example, veterans, people who are in the military can experience the same situation, but one of them will have PTSD from that situation and the other will not. I'm not saying they might have psychological issues, which are different, but they have the different perception and different physiological response to a given situation. I mean, it's hugely fascinating.
Extremely individual and extremely difficult to study, because especially for PTSD, there are also many people who are just faking it because of the benefits.
Right.
And there are some people who are hiding it because they don't want to lose their job. So if you try to do research in that field, it's extremely difficult. So this is why most of our research on stress was on animals, because there you can kind of have a controlled environment, because with humans, especially PTSD, it's extremely difficult topic.
Yeah, I completely agree. Moving on to sleep. What about sleeping glycans?
So we recently published a study with this obstructive sleep apnea.
Mm-hmm.
It's correlating with horrible changes in glycans. So what we cannot know whether this is because the apnea correlates with all the other disease predisposition elements. So it's glycans are just here kind of one of the correlated things. So it's directly affected, but definitely sleep is one of the very important components. And then again, it's extremely individual because, there are some people who are happy with 5 hours a day. Other people cannot function without 8. So it's, but sleep is, so generally it's diet, physical activity, stress, and sleep, more or less.
Mm-hmm.
And it's, we all know that. The problem is in the modern world, very few people can control diet, physical activity, stress, and sleep. So what I'm hoping is that by having a kind of a tool to monitor effects, you can focus on the most important thing. So for example, for somebody, it could be cutting down carbohydrates. For another person, it could be 2 hours of physical activity a week. For some other person, it can be just get a little bit more sleep every day, you'll be better. And you cannot know upfront. You just have to try and then see whether it works or not.
Mm. What about hormones? So like people that are taking HRT or even the contraceptive pill, how does that change things?
So hormones are extremely strong modulators of the IgG glycan. We had a properly placebo-controlled trial when there was a group of people, both men and women, where the hormones were chemically blocked. And then half of them was given either estrogen or testosterone patch or a placebo. So a woman on a placebo aged on average 9 years within the next 6 months because there was estrogen depletion.
Right.
If there was the estradiol supplementation, they did not. So estradiol is necessary to preserve this anti-inflammatory level of IgG glycan. Testosterone is also working, but through estrogen. Because, testosterone gets converted to estrogen with aromatase.
Yes.
And some people are taking aromatase inhibitors to increase the testosterone level. But if you take aromatase inhibitors, then you don't have the beneficial effect of testosterone because it's actually working through estrogen. So it is estrogen which is significantly regulating the IgG glycan, so much that we are actually currently trying to position the IgG glycan composition as a kind of indicator of the long-term estrogen level. Because when women are approaching menopause, in this perimenopause period, hormones are going up and down like crazy. So if you measure them in one point, it's completely non-informative. You have to measure for days to see the average level. And IgG glycan composition is integrating estrogen concentration for a couple of weeks because IgG has a half-life of 3 weeks. So measuring IgG glycan composition is one of the first indicators that somebody is approaching the perimenopause. So for contraceptives, I don't know. I haven't seen a strong effect. We looked into some population cohorts. You don't see the strong effect. The problem there is there are different types of contraceptives, different ratios of hormones. So we don't have really proper study with each of them. But my guess would be it doesn't have such a strong effect.
That's really good to know. And I know we're coming up on time, and I have one more question for you, Gordan. Is how do you think Glycanage clock compares to other longevity and health span clocks out there, such as telomeres or DNA methylation?
So we have so far 2 studies where the Glycanage was directly compared to other clocks. And of course, they all correlate because they all correlate to chronological age. But when we look into the acceleration of individual clocks, they do not correlate. So acceleration in glycan aging is different from acceleration in epigenetic aging. And acceleration in glycan aging seems to be more related to some kind of future, in specific study, it was future hospitalization, so some kind of a disease development. It's more reflecting the immune system. Another problem we have with epigenetic clocks is there are so many different epigenetic clocks, and they measure different things. They just have the same name, epigenetic, and and some of them don't even correlate to each other. So it's from the epigenetic perspective, at the moment, it's still very difficult to decide which test to use. And I know, for example, in this Singaporean clinic, they would measure 3 different epigenetic clocks and then take the median, which is kind of, I think the epigenetics still needs to, the field of epigenetic clock research still needs to decide about which clock to use, because of course there is a Steve Horvath epigenetic clock, which is fantastic clock. It's a multi-tissue clock. It's extremely accurate in measuring chronological age. But measuring chronological age is not information which we really need, because we know our chronological age.
Yes.
So it's a great test if you need it for forensics. It's a good in some kind of a cohort studies. But for a person, the original epigenetic clock, it's not really informative. And for the other ones, we still need more research. For telomeres, it's complicated because there are some studies showing that actually longer telomeres are the risk of cancer. Because the reason why we have telomeres is that cells do not become tumor cells. So shortening of telomeres is something which is needed to prevent cancers. And when you just measure telomeres from blood, you're not really sure whether it's a change in the composition of a blood and what is going on. So well, of course, I'm confined here. I'm the inventor of the glycan clock, so I think the glycan clock is the best. But I really think that the glycan clock is the most informative, and it's actionable. So you can, because glycans are the effectors. These are the molecules which regulate function of immunoglobulins. Immunoglobulins are important in inflammation. So what is the functional role, what is the mechanistic aspect. For all these other things, especially different epigenetic clocks, we still don't know what are they actually measuring. They're measuring some sites which correlate in populations with some events. But what does it mean for a person? We need more research. But we'll get there. There's a lot of research going on, so we'll learn more.
Brilliant. Gordan, I just want to thank you so much for coming on the show. I've definitely learned a lot, and I do hope that we can do this again soon.
Thank you for the invitation. It was a pleasure. Happy to do it.
Thank you for listening to the Functional Health Podcast. You can find links to everything that we talked about today in the show notes. If you have a second, please consider leaving a 5-star rating on iTunes. It really does make a huge difference and helps get this valuable information out and reach more people. Don't forget to subscribe so you can stay up to date and know whenever I release a new episode. You can connect with us on Instagram, Facebook, or our website, and all questions are welcome. As always, thanks to Josh Aurelia for all the editing, and thank you all for your support.